Okay, so hello and welcome to the last lecture of the African Bioinformatics Institutes and the African Plant Breeders Association Agri-Informatics series. Today we're very fortunate to have Dr. Nelson Lubanga joining us to talk about applied genomic prediction. Dr. Lubanga is a quantitative geneticist at the Institute for Biological, Environmental and Rural Sciences at Aberystwyth University. He holds a PhD in plant breeding and genetics from the University of Nottingham and has over a decade of experience spanning academia, industry, and international research, including positions at Unilever Tea Kenya, the Rosen Institute, and Doom and Orange. His research focuses on genomic prediction, breeding program optimization, and accelerating genetic gain in crops facing the challenges of climate change and global food insecurity. In today's talk, Dr. Nelson will be talking about plant breeding programs which face a fundamental challenge. They need to deliver faster genetic improvement under increasingly variable climates, and often with tight cost and resource constraints. Genomic selection offers a powerful solution, but translating prediction models into practical breeding decisions is a barrier for many programs. So this webinar presents applied genomic predictions across diverse crop systems and breeding contexts. So thank you, Dr. Nelson, and over to you.
Thank you, Samir. Good afternoon, everyone, and thank you so much for joining. So as you've heard, my name is Nelson. I'm a quantitative geneticist by profession. Currently, I'm based in the UK at IBAS, and I'll be taking through applied genomic selection, how we can use it to accelerate genetic gain in plant breeding programs. And feel free to stop me and ask any question if you have any. So this session is in 5 parts. So about 10 minutes each. We'll start with the tea breeding program. I started off as a tea breeder at Unilever in Kenya before moving to the UK. So I'll tell you some of my practical experiences and how I implemented genomic selection, its constraint, how GS can help with tea quality. Then we'll look at strategies that we can implement genomic selection in a breeding program. And then from there, we can broaden out the multi-environment genomic prediction using groundnuts, chickpea as examples, and sparse testing on how to get good predictions with limited phenotyping. So by the end, you should understand the tea breeding goals, how tea quality is evaluated, be comfortable with genomic prediction for quality traits, to know how different strategies that we can use to implement genomic selection in breeding program, and also to understand how G by E, or genotype by environment interaction, across groundnuts, chickpea, miscanthus, and also we can be able to demonstrate how sparse testing can be used in cassava, groundnut, and miscanthus. So just a brief introduction. So tea is the second most consumed beverage in the world, and is very important for most of the farmers in Kenya and also in Asia, especially India and China. But majority of it is planted by smallholder farmers and in resource-constrained breeding programs. So there's no much budget in a breeding program. And also, it has got a challenge of long generation interval. It takes about 16 years for, let's say, a trace like quality and yield to stabilize for tea. So therefore, we need strategies that can be able to help us improve tea quality much faster. So the tea breeding goals are the usual aspects. We have yield, quality, drought tolerance, pest and diseases, resistance, and adaptability across environments. So, so quality is also an important aspect because it drives the market value for different tea types. So we have green tea, black tea, oolong, and white teas all come from the same species but different in processing. And so the difference in processing and the sensory and chemical profile we are trying to select for affect how the market value of tea. in the past, people are mainly focusing on yield, but now the consumers are now focusing more on quality, and that's why we are developing high quality tea varieties. So traditional breeding relies on phenotypic selection. Um, so with tea, you can only meaningfully select once the plant has matured. That creates a delay in measuring yield, stability, and cap quality, the sensory properties of the tea infusion. So we call this the phenotypic lag, and the consequence is a slow rate of genetic gain. So you're always waiting for many years to find out if a cross was good on how it will perform. So the question we ask is, can we identify elite genotypes at the seedling stage using DNA markers? So that's the key question. So I'm sure some of us have heard about the breeder's equation. So the breeder's equation tells us the genetic gain in accuracy times the selection intensity and times genetic diversity divided by the time it takes to complete a breeding cycle, what we call the generation interval. So genomic selection gives us three levels, three levers on that equation. It can shorten the generation interval because we are not waiting for phenotypes. It lets us increase selection intensity because we can screen more candidates cheaply. And done well, it maintains or improves the prediction accuracy relative to phenotypic selection. So, in our first study, we looked at about 100 T genotypes. So these were derived from Unilever Tea Kenya clonal field trials and were evaluated at two sites. So we measured 12 quality traits using NMR spectroscopy and calculated blobs per trait, treating genotype and sites as random effects in a mixed linear model. So for genotyping, we use GBS markers. So initially we had about 82,000 markers or SNPs, and then filtered down to high quality SNPs, about 3,000 SNPs. And then we compared six prediction models. One of them was using only pedigree information, and the other five were using the marker information. So we published this paper in about 5 years ago. You can have a chance to read this paper. So, here's the predictive ability across the traits and the six models. So we had about 12 traits and six models. So if you look at the picture clearly, so the black one is the pedigree model, and the other five are the commonly analyzed genomic models. So we have the three Bayesian models, so the Bayesian A, B, C, and then BRR and GBLAP model. So by visually looking at this plot, you can see that the genomic models were almost similar, but the predictive ability varied by trait. But the pedigree model always lagged compared to the genomic model. So that's the main thing you can see from this plot. Um, so what can we conclude from this? So the performance of the genomic models was fairly similar overall with the ridge regression and Bayesian A models at the highest, followed by GBLAP model and the RKS models and the pedigree combined at about 0.5, slightly ahead of the rest. So the pedigree model was almost the weakest. And of course, as we saw, the predictive ability was varied by trait. And then, so basically that's what we kind of found. So these are kind of good prediction accuracies. So which kind of gives us an insight into how we can actually implement genomic selection in a breeding program. So then, um, so knowing that genomic selection works for T quality is one thing. So the next question is whether it actually improves the breeding program as a whole. So we simulated a full T breeding program for T. So a diploid genome with about 15, T is a diploid genome with about 15 chromosome pairs and with 12 founder genotypes producing with about 2,400 QTNs. And the trait was yield. And we also simulated about 9,000 SNPs over a 40 period that was under phenotypic selection. And then we compared about 5 different um, uh, programs. One of it was the phenotypic selection. The other one was pedigree selection program. And the other three were 3 different genomic selection strategies. So we published this in the plant genome. We can have a chance to read this paper. Um, so briefly, this is the baseline phenotypic selection program. I think most of us could be familiar with this if you're a breeder or you come across a breeding program. So, so this is a conventional phenotypic selection program. So you make crosses here. So you have 20 parents in this case, and then you get roughly about 10,000, you make about 10,000 crosses. You could get, among these 10,000 crosses, about 3,000 or 2,000 um, uh, could germinate out of that. So once you have these 2,000 seedlings, you plant them in what you call the preliminary trial. From this preliminary trial, which takes about 2 years, you select about 500 genotypes. Sorry. Sorry. So you select about 500 genotypes from this preliminary trial. You take it to what you call the advanced clonal trial. And the last stage is the elite clonal trial, which takes about, so you select about 40 genotypes from the SCT stage to ECT stage. And then from here, you release either 1 or 2 varieties from that. So that is the phenotypic selection strategy. So for the pedigree and genomic strategies, we start intervening early. Instead of waiting until the elite clonal trial stage to select parents, the pedigree and the genomic strategies select parents right at the seedling stage. So if you look at here, we have the pedigree method and the genomic selection strategy. So, and what you've done is we've shortened the breeding program by removing the preliminary stage. So you move from crossing to seedling, and then instead of preliminary trial stage, you move straight to SCT stage. So that is the main change. So we have 2 strategies here, the pedigree and the genomic. So the pedigree information, you're only using pedigree information in a model, and genomic model, you're using markers in a model. So that's the main. Okay. So the last strategy for genomic is what we call the ECTGS. So you can see here we've removed 2 strategies. We have removed the SET stage and we have removed the preliminary stage in the program. So we want to see if we shorten the breeding program and increase the number of clones evaluated at the ECT stage. Instead of 40 genotypes, we have 90 genotypes. And then we're selecting the parents at the seedling stage. What level of genetic gain are we going to get compared to the phenotypic selection stage? Phenotypic selection trial, sorry. So this is the overview of the breeding programs. So we have the 5 breeding programs. So we have the phenotypic selection program. So this is the traditional breeding program. Then we have 2 parent selection strategies. So you could select parents. You could replace all the parents, which is all the 20 parents, or it could replace only 5 parents. And then, so for this strategy, for instance, you are selecting parents at the last stage after 16 years of breeding. And then for the pedigree seed program, you're selecting parents at the seedling stage, which is at the second year of planting. And then you have 2000, all the 2000 seedlings, and then you're using the pedigree information in a model. We have another strategy, the seed GS strategy that we just looked at, constrained. So here, instead of using all the 2000 seedlings, we are constrained to use 800 seedlings because of the costs of genotyping, which is quite expensive, as we know, especially in Africa. And then for seed GS and constrained, um, especially for breeding programs that have sufficient resources, you can use this program to increase the number of seedlings. So instead of using only 800 seedlings, you could use 2000 seedlings at the you could advance 2000 seedlings instead of 800 seedlings. And the last strategy that we looked at is you've removed the preliminary trial stage and the ACT stage and increased the number of clones at the ACT stage. So that's the main difference. And then one of that is one other strategy is like you're kind of trying to replace 25% of the parents and 100% of the parents. So that's the main thing about this program. So we look at the genetic gain. In this case, genetic yield for yield will be measured over a 40 years period. So you can see that, so we have the 25% parents replaced and 100% parents replaced. So you can see that the black plot is for the phenotypic selection strategy. And then the yellow plot is for the pedigree information strategy. And then the three plots are for genomic selection strategies. So clearly you can see that the phenotypic selection performed way below the genomic selection strategies across the 40-year period. And also the pedigree information, pedigree pedigree scenario also had or came second last at compared to the other genomic selection strategy. So from this simulation exercise, you could see that the using genomic selection in a breeding program has got a long-term promise compared to using both the pedigree information and the phenotypic selection. So we also looked at the genetic variance. So an interesting thing we found that using over time, genetic variance using genomic selection strategies reduces over time significantly compared to the phenotypic selection program. So that's the key. So that's the trade-off. So which means that in a breeding program, we need to balance between genetic variance and genetic gain. So some of the conclusion is that so by year 40, all the genomic selection programs significantly outperform both the pedigree and the conventional breeding programs. So the rankings you could see was the seed GS and constraints, whereby you're using more resources. And then secondly was the seed GS constraint, whereby you're only using 800 seedlings instead of 2,000 seedlings in a program. And then the last one was the ECTGS, where you removed completely you removed two steps in a breeding pipeline. So, and then at 100% parent replacement, we are able to get about 1.7 times the genetic gain of phenotypic selection using the CGS constraint and about 1.70 times for the CGS constraint program. So the bottom line is, so the GS programs deliver substantially higher genetic gain than the conventional phenotypic selection, even under the both constrained and unconstrained budgets. So typically for programs in Africa whereby most of the resources are constrained or limited budgets, genomic selection can actually help because we kind of we benchmarked the cost of implementing genomic selection via use compared to the one when you use only a phenotypic selection program. So, and we found that we can actually implement genomic selection perfectly in this case for tea breeding program. Is there any question before I move to the next topic? Good. So now we now move beyond a single crop and single environment. So G by E means genotype ranking can change from one environment to the next, which could complement the selection process. And the key question here is, can we use genomic information to predict performance in environments we haven't even tested yet? So that's the main question, especially for a breeder. So we do have several cross-validation methods, or what we call the scenarios that we use to simulate different breeding scenarios. So CV1 will predict newly developed genotypes that are unobserved in every environment. So we have CV2 that mimics the sparse testing, what you call the sparse testing. So meaning the genotypes and environments are both known individually, but not in every combination. And then CV0 predicts genotypes that we've already tested, but in a new environment. And then CV not not is the hardest scenario, predicts genotypes we've never tested in any environment. So the environment and the genotypes are all new. So we are kind of trying to test both. So these are the unique scenarios we test that are unique to breeding programs or breeders. So the first study we did was for groundnuts, and I was collaborating with ICRISAT. So we had 192 genotypes across 4 locations in Uganda. So 4 locations, 2 locations in Uganda and 2 locations in Malawi, a place called Serere and Nakabango, that is in Uganda, and Chitala and Chiteze in Malawi. An experimental design in alpha lattice design with 2 replicates. And we had 3 main traits that we are looking for. So pod yield, seed weight, and 100 seed weight. So we had about 38,000 SNPs. And the first stage was to estimate what you call the blues within each environment. And then we used those blues in the models that we fitted. And I had 4 different models. So the first model was the baseline model, which was only the environment and the genotype. The second model, you fit the environment, the genotype, and then the genomic. So the genotype is the line or the variety, you call it variety or accessions. Genomic is the markers. The third model, you have the environment, genotype, genomic, and G by E. So you're kind of increasing the complexity of the model. And the last model, I wanted to see the effect of incorporating significant GWAS markers as fixed effects. So what I did was to fit the environment, genotype, genomic, G by E, and then significant markers as fixed effect in the model. So you do the GWAS, then you select the markers that are significant. In this case, I used the Bonferroni correction. And then I was looking at 4 scenarios. So that is CV1, CV2, CV naught, and CV not naught that we looked at, and then 5-fold cross-validation across 10 replications. So if you're interested, we publish this paper again in the plant genome. You can have a look at this paper if you're interested. So here is the results. I will not go into details, but just an overview. So this is CV1, CV2, CV not naught, and then CV naught. So we can see that the predictive ability was high in CV2 and CV naught, and lowest in CV1 and CV not not as we'd expect. Um, so the main conclusion or the key findings from these results was that the CV2, the sparse testing, the scenarios simulating the sparse testing, predictive ability was the highest overall. And then all the genomic selection model under this scenario beat the baseline model for pod yield and seed weight, but not 100 seed weight. And then, of course, under this scenario, adding G by E or the G was significant markers added little impact on the predictive ability. And this is because we already have more information using the sparse testing. So thus the model were almost the same in terms of predictive ability. So under the CV1, whereby you are kind of trying to predict newly developed genotypes, the M4 was consistently the best model, and of course M1 was the worst. And incorporating G by E beat the main effect model, and of course adding significant markers also improved the predictive ability across the trait. So under CV1 method, you kind of benefit by kind of including G by E and the significant markers as fixed effect in the model. So CV naught was almost similar to CV2, whereby you're trying to predict genotypes in new environment. So the simpler model outperformed the best model, that is the most complex model, that the G by E and model incorporating um, the significant markers. So they are almost the same. The CV naught, and the CV2 method were almost the same. So, and the CV naught was almost the same as the CV1 and these are the most complex scenario. So where you are predicting new genotypes in new environments. So the marker fixed effects model, which is the M4 model, outperformed the baseline, and the G by E plus significant markers also beat the main effect model for pod yield. So the bottom line is, so G by E remains a critical challenge for estimating breeding values across the environment, especially in this region. The strongest predictability under CV2 confirms that more information on non-genotypes and non-environment generally helps. So significant markers combined with G by E were the most valuable precisely when predicting a non-genotypes or a non-environment, that is CV1 and CV naught naught. I don't know if we have any question up to that point. Okay. Another scenario of multi-environment genomic prediction was I have been looking at chickpea dataset obtained from ICRISAT. I'm always grateful with ICRISAT. I have some collaboration with ICRISAT. So I had 209 chickpea genotypes across 9 environments in India. And the genotyping method we used was the whole genome sequencing dataset. And so we had 2 main panels. So the genome-wide panel had about 400,000 markers or SNPs, and the LD-based tagging, so whereby I kind of selected the markers based on the LD. So by doing that, I only remained with about 9,000 markers. And then, as usual, the first stage of the analysis was to estimate the blues within each environment. And I looked at 4 cross-validation strategies we've just looked at, the CV1, CV2, CV naught, and CV naught naught. And we looked at 4 different prediction models. The M1 is the baseline model, that's environment and line. The second model is the the main effect, genomic main effect model. So we are increasing the complexity. So M1, environment plus line, then plus genomic main effect. M3, we incorporate G by E in the model. In the last model, you incorporate GWAS significant markers. But in this case, I was using the FDR 5% across all the environments as the selection criteria. So this paper, it has been accepted by the theoretical and applied genetics. So it will be published anytime soon from now. So I'm not able to share the paper now, but in the next couple of days or maybe 2 or 3 weeks, it should be published.
Nelson, we have a question in the chat from Johannes.
Uh,
Johannes, if you like to unmute and ask your question.
yeah.
Johannes, would you like to go ahead and ask your question, or I can read it out for you? Okay, Johannes' question is, which genotyping method did you use? Whole WGS, whole genome sequence, ddRAD, .seq, or something else? Will the
For
genotyping method and depth have an impact on the result?
So this one, you can see I use the whole genome sequencing method. So this is whole genome sequencing. And the initial, if I can just give you a break, like uh, a snip through. So all the markers were about 3.9 million markers. So I had to kind of use the standard quality control methods to come up to about 409 SNPs. And then for LD tagging, the main aim of the study was to check, okay, um, I mean, this has an impact in a breeding program. So if you are using 400,000 markers and someone else is using about 9,000 markers, there's an impact in terms of cost in a breeding program. So if the people working if you send your samples to a genotyping company, you tell them you want about 9,000 markers, the cost will significantly reduce, and also the computational time of analyzing the samples will be much less than someone who's using 400 markers. And I'll tell you, because I was fitting these models myself, it takes too much time to use 400,000 markers, SNPs, compared to about 9,000 SNPs, which you can do it locally in your computer compared to 400,000 markers. So, so basically, to answer your question, I used whole genome sequencing data. But the other props I've discussed before, one I used GBS, the other one I used RADseq markers. But for this one specifically, I used whole genome datasets because that's what was available in that particular breeding program at EquiSearch. I don't know if I've answered your question. Okay. Um, so if you can look at this slide, so this slide shows the predictive ability across the 4-cost validation scheme in chickpea. Okay, as expected, the predictive ability was higher under CV2 here, as you can see, was going up to 0.8. Sorry, because the slides are kind of small, but you can see that it was going up to 0.8. So CV2 had the highest predictive ability scenario compared to, okay, CV2 and CV naught had the highest predictive ability compared to CV1 and CV naught naught, because these are the most difficult scenarios in a breeding program to predict. And looking at the LD panel, so the LD panel was performed almost similarly at the CV naught and the CV2 and CV naught naught. But now, if you look at the CV1 and CV naught naught, is that the LD markers performed significantly lower compared to the genome-wide markers. And has got some implication which we can look at in the next slide. So 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 remember so 9,000 about 9,000 markers is about 3% of the markers that we use for the genome-wide uh, markup panel. So by using the 3,000 markup, 3% of the markers that were used for the LD panel, the predictive ability was almost the same for the CV2 and CV0, meaning that if a breeder is targeting those scenarios, you can actually significantly reduce your cost of genotyping and also the computational cost if you're targeting CV0 and CV0 and CV2 scenarios. But, and this is because you have more information at the sites. So that kind of helps in your prediction scenario compared to the most complex models like CV1 and CV0. So for those ones, the whole genome markers perform much significantly better than the LD panel. So that was the main conclusion. So using the LD panel, the reduced um, markers, and in this case, we use the LD tag method to kind of reduce the markers. You can actually reduce the cost of your breeding program. If you're targeting the sparse testing scenario, which is CV2, or the CV0 scenario, whereby you already know the genotypes, or you evaluated the genotype, but now you're trying to predict them in a new environment. Then, lastly, I'll go to something I'm sure some of us are practicing back in Africa, the sparse testing. So basically, we would want to release stable genotypes that outperform the current elite materials across a broad range of environments, which means that we have to conduct the multi-environment trials. But in most cases, the budgets don't allow because assuming you have 1,000 genotypes and you want to evaluate in about 10 environments, that will mean like you have to design an experiment in each environment with replicates, and then if it's, let's say, RCBD or alpha lattice design or augmented design, whatever design you're using in a breeding program, but then you have to replicate that in a different environment also. And that's very expensive to do. So recently we have people who designed what we call the sparse testing method, whereby you don't have to test all the genotypes in each environment. You can actually spread out all the genotypes across the environments. And then we use what we call using the variance covariance matrix. You can predict the genotypes that were not tested in each environment using this technique called sparse testing. So basically, you identify a good genomic prediction model incorporating the G by E for use in kind of predicting within environment genotypes that were tested elsewhere. So that's what we call sparse testing. So in a study we did with cassava, so I was collaborating with this with IITA in Nigeria. Um, Again, I was kind of grateful for IITA. So they provided about 435 genotypes, which were planted in alpha lattice design and two replicates. So the 5 environment in Nigeria with different. So basically, there were 3 different locations, and um, um, so what you call is site by ear combination. So in total, we have 5 environments, and I was looking at 2 traits, that the fresh rutile and dry matter. And then, as usual, the first step is always to estimate the blues. I like using the blues because I don't have to. Those ones who attended Lipka's lecture, we're talking about the blues and the blabs. So it's better to use the blues so that you don't regress, sorry, you don't shrink the values twice. So if you have blab, you'll be forced to do what you call the regressing the values back to the original values. So I encourage you to be using the blues before you proceed to genomic prediction. So in this case, um, we use a different genotyping platform, which is DATSIC. That's what they had at their bridging program. And we had three models. So the models have been using the baseline model, the model incorporating genomic effects, and the last model incorporating the G by E effects. So some of you might be wondering why I'm using the baseline model. It's always good to check if your model is doing much better than the standard. So the standard is like you don't have genomic information. So you have to compare, is your model doing better? Is the markers actually helping? Because it's been proven that the markers should help in improving the prediction accuracy. So for those of you who are interested in this paper, you can check um, here. So we developed sparse testing designs. So we had what you call the non-overlapping and overlapping allocation design. So non-overlapping means that you are testing all the genotypes in all the environments, but you are not replicating any. And then overlapping design is that you have, okay, completely overlapping design means that you are replicating all the genotypes across all the environments. So in this case, we started with 87. So if you have 435, you divide by 5, you get 87. So meaning you're planting 87 genotypes in each of the environments. And then, so how to design is we are kind of reducing the genotypes gradually until you have a fully overlapping allocation design to kind of test which design is better. Is it better to completely replicate all the genotypes across the environments, or is it easier to use the non-overlapping genotypes? And the thing is, so using an non-overlapping genotype becomes good, especially at the earlier stages of the breeding program, because in most cases, you don't have enough materials to replicate across, let's say, 5 environments or 10 environments. So if you have a representative, or if you can completely not replicate your genotypes, then that could prove good in your particular breeding program. So this is how the results were for cassava. So you can see, so here we are moving from completely non-overlapping, so you're not replicating any genotypes, to completely overlapping. So you are replicating all the genotypes across all the environments. So you can see that from these results, you could actually get much better prediction accuracy. So this is the y-axis is the prediction accuracy or the predictive ability, and the y-axis on the x-axis is the configuration, the allocation configuration. So this is so 80s. Just move back. So 87. Give me a sec. So 87. So 87 0 means 87 non-overlapping genotypes and 0 overlapping genotypes. And then the last one was 47. So 47, now all of them 47 overlapping genotypes and 0 non-overlapping genotypes. So that's what it means. Um, yeah. So and sorry, I forgot to mention that we're also looking at apart from predictive ability, we're also looking at the mean square error. So you'd want a model that gives you a much smaller mean square error compared to a model that gives you, a highest mean square error. So it should be the opposite. So as the mean square error reduces, the prediction accuracy should increase. So that's what we are kind of looking at in this experiment. So we find that the most complex model in this case, the model incorporating G by E, performed much better than the main effect models, that is the M2 with only genomic information and M1 that did not have genomic information across all the allocation designs, with predictive ability highest under the fully non-overlapping designs and the lowest under the fully overlapping design. So that's what we found in this experiment. And then the mean square error also showed a similar pattern. It rose as the overlap increased. And the most complex model, that the model incorporating G by E, consistently had the lowest mean square error. And then the larger training set, that is 87 compared to 47, genotype per environment improved both predictive ability and the mean square error across the three models. So in this particular breeding program, incorporating G by E was important because you could see that it had a better predictive ability across all the allocation designs compared to the main effect models. That is model 1 and model 2. And for these cassava genotypes, you do not have to replicate a lot of genotypes across the environments. You could test as many genotypes as possible across all the target environment. And if you have if you decide to kind of incorporate some replicates, it's okay, but still you can get some very good predictive ability by just using non-overlapping genotypes across all the environments. So that is what we found from this study. So another study is we were looking at is for miscanthus. Miscanthus is a perennial bioenergy crops. So in this case, it's planted in large locations in the UK here and some other locations in Germany. And miscanthus is a bit of a tricky crop to work with because it has got many species within it. So in this case, I did um, my own population structure analysis and identified three main species. So we have the Miscanthus sinensis, Miscanthus sacroflorus, and the interspecific hybrids within the trial. And because I had limited materials, so I had to use all the materials. So because if I only selected one species, I would only have very few genotypes to use, and I didn't have the luxury to only use that. So I was looking at 5 environments, that is year-term sites, both in the UK and in Germany. And then I was looking at 3 traits: dry matter, fresh weight, and moisture content. Um, and then I use the DAPC method, that is the discriminant analysis of principal component, to identify the 3 genetic clusters, and which I confirmed match the taxonomic groups. And as usual, I use the 3 different models. So model 1, 2, 3. So 3 is a complex model incorporating G by E in addition to the genomic. Maybe some of you could be wondering how I fit the M3 method. So I fit using what you call the Hadamard product, which is a cell by cell multiplication. So you have the markers and then the environments. Then you kind of fit that in a model to kind of incorporate them in do the prediction. So then I had 125 genotypes, and then as usual, I did the non-overlapping and overlapping ratios, just as I did for cassava. And then I did the prediction accuracy, which I can show you. Before I move to the sparse testing design for miscanthus, I wanted to see the effects of the G by E using the simple methods like the CV1 and CV2 methods. So you can see that the incorporating G by E really improved the predictive ability for the CV1 method. For the CV2 method, which is a sparse testing, we already have more information. So yes, we had some improvement in predictive ability using the G by E model, but it was not as significant as under CV1 scenario. So this is the predictive ability across the sparse testing designs. So this is the 100% non-overlapping or 0% overlapping genotypes. Um, so you can see that. Sorry. So this is the 100% overlapping, 100% overlapping and 0% non-overlapping, and then this is the you kind of using percentage. So this is 20% non-overlapping, like that all the way. So and you can see that um, the models performed better when the genotypes were um, had 0% um, overlapping. So this title is a bit confusing. So 0% non-overlapping 0% overlapping had the highest predictive ability compared to 100% overlapping. So that's the same results we found for cassava.
Nassan, we have a question in the chat from Abhinav.
Okay.
Abhinav, would you like to unmute and ask your question? Okay, if not, he's asking how to check which GWAS models gives false positives versus true positive SNP trait associations. So I'm guessing, how can you reduce your amount of false positives versus your true positives? I guess is the question as well.
Okay, so that's a good question. So for my GWAS analysis, what I was doing is like I would take like the main models like the Blink and the Blink and Palm CPU, for instance, and then I'll estimate I'll do GWAS within each environment. Then I'll try to kind of say that the genotype that did sorry the hits or the markers that did better across so assuming I have done GWAS across each environment, then I've found significant markers within each environment. So what I'll do is I'll compare them and select only those markers that appeared in both, if the environments are true across both environments. I don't know if that makes sense. So at least I'm sure. Um, so a marker that has, if a marker that was significant in, let's say, environment one, also was significant in environment two, then it means it is more highly likely that that hit is significant. So in this case, I'll pick that marker and fit that in the model. So for the people who want to kind of move further to, in a typical GWAS study, they want to, let's say, do candidate gene analysis and things like that. So the other ways they can select those markers and then, let's say, map it to kind of the published reference genome and kind of try to select which markers, which genes are kind of associated with those traits. Yeah. So for me, what I'll do is what my method is I do GWAS with different models, then try to select um, the markers that were significant with both models. And if I'm looking at different environments, the markers that are also significant across all the different environments, then you start in for further analysis. So that's how I do it. Yeah. So I'll not dwell much on this Miss Cantor's work because it's almost the same as what we found with cassava, that increase using non-significant markers, non-overlapping genotypes across the environments is much better than um, using only overlapping genotypes across all the environments. So lastly, we did sparse testing in groundnuts. Again, I used materials from AgriSearch, which I was grateful for. at the 4 environments in Uganda and in Malawi. So for the sparse testing designs, we also developed complete non-overlapping and complete overlapping designs across the locations or the environments to kind of see the impact of how you can allocate the genotypes. And then we used 3 different methods, 3 different models again to kind of test the baseline model and the genomic main effect model, and then the G by E model in addition. So this one, as soon as yesterday, I submitted this paper for publication, so it's not yet available for reading. But now, for this one, it gives a bit of different results. But definitely, you can see that the most complex model was doing much better than the main effects or simple model. So again, for this particular population, using the G by E model was much more beneficial compared to using the main effect models across the allocation designs. So, and then the most important part is that if you look at here, you can see that there was a bit of stability in predictability up to about this allocation design. So meaning that in groundnuts, if it would be recommended to have some replications across environments because, and especially if you want to maintain the reliability of the results, or you want to have some confidence, it's to be important for you to kind of have more replicates, not so many, but kind of some representative replicates. I saw some breeders using what you call the checks to kind of replicate across all the environments. So that would be more beneficial for the groundnuts breeding program. Yeah, so we've seen that the M3 model, the most complex model, was good and was consistent. And then the predictive ability was highest from the fully non-overlapping through to moderate overlap, declining towards the fully overlapping design. And the environment also mattered too. One of the environments called Serere gave the highest predictive ability. Chitala, which is gave the lowest predictive ability for those overlapping designs. So the bottom line is, so sparse testing with G by E modeling reduces phenotyping effort without sacrificing predictive ability for pod yield and seed weight, although the model choice should be trait dependent. A fixed 192 plot design gives a 4-fold reduction in phenotyping, or 4 times the genotypes under the same budgets with as few as 16 genotypes, 16 overlapping genotypes per environment needed to sustain the connectivity. So the most important thing for sparse testing is how connected the genotypes are across the environments. So that is the end of my presentation. We have some resources that I've just shared with you. Some of them are under review, but yeah, I don't know if there's any question or any suggestion.
Thank you so much, Nelson. I see we have 2 questions in the chat. One is from Arnaud. Arnaud, would you like to unmute and ask your question?
Yes. Hello?
Hello, yes.
Can you hear me?
Yes.
Okay. Thank you, Dr. Nelson, for the nice presentation. I'm currently working on a manuscript, a GWAS and GP manuscript. And I have used blues for both of them, for GWAS analysis and GP analysis. For the GP, I try to, I choose the RR block model for GP. Then during the analysis, I use all the significant from GWAS with RR block of the same model and all the SNPs with the same model. And I realized that the prediction accuracy for the model, including the significant SNPs, were better than the prediction accuracy for when we have all the SNPs. And let me also remind you that for the GWAS analysis, I used both GAPIT and MRMLM method. And now, and however, in the literature, I realized that many papers, they use blob for genomic prediction analysis. Like instead of using BLUES to extract, they use blob. Like for the extract blob for to run the GP. So I want to know in which case do we prioritize blob for GP?
Yeah, so as I explained earlier, I would advise you to please use your BLUES because
Okay.
you see, when you use blobs, it means you've already shrunk your estimates because those are estimates. So you've already shrunk your estimates. So if you take those estimates again and go and fit the model in a genomic prediction model, you're kind of shrinking them again because genomic prediction makes those predictions. So we have what we call double shrinkage.
Okay.
So, but for BLUES, you're not shrinking. So you're using like what we call almost like the absolute values to fit in the model. So
Okay.
I'll advise you to use the BLUES as you're using and not blobs.
Okay, thank you.
Yeah.
Great, thank you. We have a question from Enoch. Enoch, would you like to unmute and ask your question?
Okay, thank you very much, Nelson. I just wanted to find out on the Miscanthus paper, I noticed that the fresh weight, dry weight
Yeah.
were slightly different from the moisture content when you
Yes.
did sparse testing. So I was asking myself, is it an issue of the heritability or it's something else? And then I was also looking at when I'm looking at sparse testing, we have different sites, probably with different uh, endogenous errors because maybe you have different teams, we have slightly different maybe equipment to take the to record the phenotypic data. Would you think maybe these are factors that could impact on the accuracy of the model?
Yes. So, okay, first of all, to answer your question, I've just extracted those results. So to answer your question, so 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 moisture content is a different trait to compared to dry matter and fresh weight. So to be honest, so basically fresh weight and dry matter is the same thing. So you get the fresh
Yes.
weight and then you get the dry matter from that.
Mhm.
So that's why the trend for moisture content could be the same. And even the heritability estimate. Sorry, I didn't present the heritability estimate, but they should be the same, almost the same, because it's the same starting material. So moisture content is a different trait. So you expect kind of different configuration compared to let's say the dry matter and fresh weight. But again, also, of course, factors like heritability will kind of impact on the predictive ability. So if the heritability is high, it means that we have very high precision, or the genetics is kind of more likely to explain more of the trait variation compared to if the heritability is low. It means that we have more errors, or like the traits are kind of less heritable. So, okay, one of the things of increasing heritability is, let's say, making more replications, replicating more of your trials. So if you replicate more, it kind of means that you have more confidence in your estimates, and definitely your stability will be much more higher. So to answer your question, if you look at this, so the heritability would kind of, if you're trait heritability is higher because it's kind of heritability is a trait dependent scenario. Sorry, population dependent scenario, and also trait dependent. We could have the same genotypes with different traits. You don't expect them to have the same heritability estimates. So within each trait, the predictions will be much different based on the heritability estimates and all the other factors like is the training population, for instance, similar to the prediction population and things like that. So sorry, I didn't present the heritability estimates, but
Yeah,
that's what
while you are still there, Nelson, I was looking at this trait, the moisture content, and I was asking myself that, look, this is the difference between the two other traits. So why would it be so different? Because when you look at the dry matter and the fresh weight, then the difference will give us an indication of the moisture content. Then why would it behave so differently?
so okay, that's a good question. So it behaves differently because so moisture content and fresh weight are these are yield related components. I'm not a breeder, but these are yield related components. So this is what is more important for breeders when they're trying to, let's say, estimate the yield of miscanthus, for instance. Moisture content is more like a quality trait. So it's kind of different from fresh weight or dry matter content. So basically, you don't expect it to behave the same way because that totally different traits. It's like, let's say, if you say something like in people working with, let's say, uh, crops like, let's say, in maize, the quality of how the maize is testing or the organoleptic properties is different from the yield, and they're most likely different in terms of the like so let's say in a breeding program, if you're trying to improve, let's say, yield, would be different from if you're trying to improve the quality of the of that particular trait, sorry, crop. So the thing is, the traits are different because they are different, and what some of the things we do in breeding is that we'd want to, let's say, improve them simultaneously, and that's what you call the multi-trait index. So if I was more interested in this trait, moisture content, and I want to improve it with these other two traits, then I would, let's say, develop a selection index to see if I can improve both simultaneously because they behave differently compared to these other two traits. So maybe if you want, you can take the discussion. You can write me an email, and then you can discuss more if you would like to.
No, it's fine. It's
Yeah,
all right.
yeah.
Yeah, I think my other question was on the different environments and probably the use of different equipment, especially when maybe when the data capture is not like mechanized, when it's still subjective,
Mhm.
you'd still expect to that to bring in some source of variation.
Yeah.
So yeah.
Yeah, that's a very also important observation because I'll tell you for free for groundnuts. I'll show you for groundnuts. So for groundnuts, we had four different environments. So we had to drop some traits because the heritability was very low, and as
Mhm.
you'd expect, someone who's measuring some traits, even yield trait, let's say in Malawi, is a different person measuring the yield trait in Uganda, for instance. So one thing is like we'd find that in some traits, you find like some traits had almost zero heritability, while in the same trait in a different environment, the heritability is
Mm.
like 0.8. So this stems to the fact that because I'm not a breeder, I'm more like a quantitative. So I depend on the breeder to tell me, okay, do you think you did this analysis, you did this data collection properly, or what happened? Because I mean, if in one environment, the heritability is 0.8, and in the other environment, heritability is almost zero. It means there's a problem. So I cannot use that data set, right? Because normally we advise if you have a heritability of 0.2, there's no point using that um, data because it will distort your rankings across the environment. It's recommended you use at least 0.2. We accept that we do have variations between the environments, but just try to maintain that you have a heritability of about 0.2 at least within each environment. Then you can now fit a model. So, but the most important thing is for you to estimate heritability across the environment to kind of look at how the quality of your data is, because heritability is the first check for your heritability. For your prediction, for your accuracy of your results. So if your heritability is poor, it means generally it could mean two things. Generally, that trait is poorly heritable. Secondly, it could mean there's some errors in your data collection, and you need to check. So, and if the trait is very low, 0.2 and below, for me, or from the standard practice, that you remove that trait from your analysis, because even assuming you submit that paper, people ask you, why did you include that trait when clearly it's not heritable? The heritability is very low. It's more like an
Yeah,
indication of some errors in your data collection.
it's all right. Thank you, Nelson.
You're welcome.
Okay, thank you. I see Kwame has his hand raised. Kwame, if you'd like to ask a question.
Hi, Nelson. Thank you for such a nice presentation. I just wanted to ask about the overlapping and non-overlapping of genotypes. I think you can even look at your previous slide. I just wanted to know, what do you think is the optimum number of genotypes to replicate across environments? And also, do you replicate the same genotypes across all the environments for sparse testing? I'm still new to it, so yeah.
No, it's a good question. So the thing is, it's okay to answer your question. It's trait dependent. Sorry, it's crop dependent. So, with my collaborator in the States, we've been doing this with my collaborator in the US, at the University of Florida. He's also looking at different crops. So for me, I was looking at different crops. So what we found is that for my crops, um, for cassava in this case, using non-overlapping genotype, like you're not replicating any genotype across the environment, you get much higher predictive ability compared to if you're only using overlapping genotype across the environment. And the basic answer for this is because, okay, for cassava, and my friend um, used sugarcane, and also I used miscanthus. So these three crops are what we call the clonal propagated crops. So for these clonal propagated crops, there was no point of replicating large numbers of genotypes across all the environments. So that is from our analysis, that's what you find out. So I mean, you'll get better benefit by only using an overlapping genotypes across the environments. And this is mainly because, I mean, you're using more genotypes. In this case, there's more genetic diversity available within the training set for you to make predictions. So that's the most likely explanation for that. Secondly, for other crops like, let's say, groundnut, this is miscanthus. For other crops like groundnuts, like here, you can see that actually adding some replications across the environment could be beneficial because there's a bit of a steady predictive ability across the environment up to a certain level of overlapping stroke, sorry, not overlapping stroke, overlapping design. So including some replicates within your breeding population or your trials could help in the long end for this particular crop, groundnuts. Also, my friend was working on soybean, which is also a legume. He found the same thing, like including some amount of replicates across the environments would actually help improve the prediction ability of that particular crop. So so to answer you is kind of trait dependent. And also, why this design is important is because sorry. So why these designs are important is because sometimes earlier in the breeding program, you don't have enough materials. Let's say at the seedling stage, you only have one copy. You don't have more replicas to kind of plant across the environment. So most breeders would throw away those materials, or they'll have to wait until they have sufficient materials to replicate before making a decision. But you see, with sparse testing, you don't have to wait for long. You could subdivide those materials into 3 or 4, depending on the resources you have, and just evaluate them and make your prediction as soon as you can. Then you advance your breeding program. So that's why sparse testing is kind of a really promising approach, not only for resource limited breeding program, but also the efficiency. Because I mean, you already have information that you could get in other environments, compared to only having information in one environment. So I don't know if I've answered your question.
I think partly, but just one more thing. I just wanted to ask then, how do you model the G by E if there is no replications across the environment?
So you use a model incorporating G by E. So okay, picture about this. So picture this. So you have, let's say, 100 genotypes, and you have 4 sites. So those 100 genotypes, you divide by 4, you get 25 genotypes. So those 25 unique genotypes, you plant them in one site. The other 25 genotypes, you plant them in a different site, like that. And then the other one, all those 4 different sites. Then you genotype all those materials. So you have SNPs, right? So when you have the SNPs, then you have this, you can actually fit this model. You fit this model. So you have, let me put this model that I fit properly. Yes. So you fit this model. You have the environment. So like, let's say environment 1, it could be Uganda. Environment 2 could be Zimbabwe. The other environment could be South Africa, like that. So those are the environments. Within those environments, you have the genotypes. We fit the genotypes, and then we have the markers. So you kind of build a kernel for the markers to kind of fit in. And then you also do what you call the G by E in the model. So G by E is what I was saying is the Hadamard product. So you're kind of multiplying the cell by cell product of the markers and the genotypes across environments. So that's the key feature. So you're kind of multiplying the markers and the environments across the environments to kind of give you uh, the covariance matrix of the environments and the covariance matrix of the genotypes.
Okay, so it's the marker information that helps you to model the G by E.
Yes, correct.
Okay, no, thank you so much.
But also, if you have a good pedigree information, you could actually use pedigree information instead of the markers if you don't have the money for genotyping. However, the markers are much more preferable than the pedigree information. Because the pedigree information, because of what you call Mendelian sampling. So because of Mendelian sampling, the pedigree information is much more reliable than the markers. Yeah.
No, but thank you so much. That clears.
Yeah, you're welcome. Thank you.
Great. Do we have any questions for Dr. Nelson? If not, if you could give him a big round of applause, virtual applause. Thank you so much, Nelson, for your great talk.
Yeah.
And thank you everybody for joining us for the last of this lecture series. And thank you. And thank you to our speaker, Nelson.
Thank
See
you
you.
so much. Bye-bye,
Bye.
everybody.